This protocol presents insight into algal blooms and how to monitor toxic algal species effectively. The procedure helps provide an early warning of algal blooms to protect the community from associated damages. Metabarcoding analysis can detect many species in a sample at once, but it requires complicated procedures. This protocol visually explains each step to help minimize human errors during the analysis. The microscopy procedures will be demonstrated by Henry Cameron from Antofagasta University, sample collection by Jonnathan Vilugrón from Instituto de Fomento Pesquero, sample pre-treatment by Karen Vergara from Los Lagos University, and the metabarcoding analysis by Ignacio Rilling and Marco Campos from La Frontera University. Begin by collecting 3 liters of water sample from a target spot. Filter 1 liter of the water for 16S rRNA analysis through a tandem filtration to separate the free-living and attached bacteria. Next, filter another 1 liter of water for 18S rRNA analysis through a single filtration with a 0.2 micron membrane. Then, using sterile surgical scissors, cut the filtered membrane in half, wrap one half with aluminum foil for storage, and with the other half, proceed to DNA extraction with the Kellex method. For microscopic analysis, transfer 1 milliliter of the water sample onto a 1 milliliter grid slide using a pipette. Observe the sample under a microscope and record the names and quantity of the phytoplankton species. Clean pipettes in the laminar